And we're going. So 5.5, it's looking at bulk transport. So if you buy in bulk, where are you buying from? Costco, right? You buy in large quantities. So bulk transport is when the cell wants to either exiting, send stuff out of the cell in bulk, or endo, you want to bring cells in the cell, that is, stuff. You bring it in bulk. You bring a lot of things at once. Okay? Now, I want to review like what kind of things can go through a lipid bilayer just by like normal simple diffusion. So here's my lipid bilayer, right? And I'm not going to draw a whole cell membrane because that would take too long. But what kinds of things can just like go through the cell without any energy, without any help? They just go right through. There's two properties that would cause them to be able just to go in unimpeded. Yeah, go ahead.
Non-polar molecules can go through?
Non-polar, that's a huge one, right? Because these tails, these fatty acid tails, are non-polar. What other things? The other one's like a little more obvious. Imagine I'm 10 foot, I'm trying to like get through that door. You know? Antigone? Yeah?
Smaller molecules?
Yeah, you're smaller. So, and you really need both of these. Now, if you're something like water is small. Like H2O is a pretty small molecule, but it's polar. So water can still like kind of like, think of it like this as a crowd. Water can, or like the hallways at McLean, right? It can kind of make its way through, but it's not coming through very efficiently because it's polar. Whereas if you're non-polar and small, you're almost like a ghost, just like passing through the hallways. You know, you can go through pretty quick. So that's why water then would need like some sort of, we call it the aquaporin, that channel protein. Anyways, just kind of a review of what can bring, like what you need to get into a cell. Yeah, some of the large things you might bring, it might be like polysaccharides, proteins. Anyways, bulk transport requires energy. And when I say energy, we're talking about ATP. Okay, ATP. Okay, let's get into an example. Oh, before I get into an example, what do things, when things normally come in the cell, what do they normally do? When I said like passive transport versus active transport, so like passive versus active, what was the main difference? Do I remember? Yeah, Sean, go ahead.
Moving down the concentration gradient?
Down your concentration, that'd be passive, right? You go from high to low, and that just happens naturally. You don't have to put in energy. But if you're in active transport, you're doing the opposite. Now you're going low to high. Okay? Now if you Google exocytosis, endocytosis, what kind of transport are they? Google will tell you that they are active transport. And because active transport, to go from low to high, you have to use ATP. Now, if you look at one of the Mastering Biology questions, it'll, like if you say like endocytosis, exocytosis are always active transport, it won't tell you that it's correct. I don't personally care, but what they're getting at is, when I show you some of these examples of exocytosis, endocytosis, you don't always have to go from low to high. Sometimes you're going with the concentration gradient, but you still, because you're bringing things in bulk, you still have to use ATP. I'll give you an example to explain a little more what I mean, but I just want to preload that, I guess. Okay, so let me show you exocytosis. So here's your example of exocytosis. Typically, exocytosis is waste. Like if you're your average cell that isn't really like involved with like making things, you're usually doing exocytosis to get rid of waste. Where you're going to have a vesicle. Remember, a vesicle is just a storage container that's got a lipid bilayer around it. And then these blue dots would be the waste. It's then going to fuse. So here's your vesicle. It then fuses with the cell membrane. And then it's going to release the waste into the cell. And then that cell membrane that was forming the vesicle becomes, well, the membrane, not cell membrane, the membrane that made the vesicle then becomes part of our cell membrane. I mentioned that a little earlier in the class, where like we then get to add this cell membrane to our this membrane that made the vesicle into our cell membrane. So exocytosis will increase the surface area of the cell membrane. Okay, so that's an important thing to know. When you do exocytosis, you are adding membrane from the vesicle into our own cell membrane. So if I were to do endocytosis, where I'm doing the opposite, I'm bringing stuff into the cell, I would then decrease the surface area, right? You're doing the opposite. Now you're removing membrane. And so because in most cells, this is kind of like the process is balanced out. You're releasing and adding at the same rate, the cell membrane will stay relatively the same size. Now, some other examples of exocytosis. Maybe if you're like in the pancreas, you might make insulin. Well, you're gonna these blue dots wouldn't represent waste; they would represent insulin, a protein that's useful for bringing in sugar into the cell. So it's not always waste. Another example of exocytosis would be in a neuron. So I want you to know this example. I think it's kind of useful. You don't have to know the exact structure of a nerve cell, but you know this axon, this will connect up with what we call a cell body, and then you'll have these dendrites that come off here. These are the dendrites. The axon is where you're going to send a message, and then that message will then go to the dendrite of another nerve cell. So then that dendrite, there's a bunch of other dendrites here. They then connect up to the cell body, and then this nerve cell would have its own axon to send another message. Anyways, inside these vesicles, this wouldn't be waste, these would be neurotransmitters. Neurotransmitters are how nerve cells will send messages to one another. And so through exocytosis, we would release those neurotransmitters into, anybody know what this is called? You don't have to know it, but I'm just curious. Kai? No? This would be the synapse. There's another, like a two-word name for it. Ashley?
Is it just the gap?
It is a gap, but there's like a yeah, go for it.
Synaptic gap?
Synaptic. Riley? Synaptic, not. I mean, it could be junction. Maybe they call it that, but Will?
Synaptic cleft.
Synaptic cleft. The best diss that my best friend's dad told me this the synaptic cleft. You know, if you wanted to diss somebody, you would say, "Hey, so and so has a really large synaptic cleft," meaning it takes a while for information to travel to them. You know, they don't really pick up on it very quickly. Anyways, okay. Yeah, so don't even know the details of how the neuron works. Just know that's an example of exocytosis. All right, so that's really it for exocytosis. Remember, this process would require ATP. That, just to be clear, in order for these to fuse and for us to spill out those molecules, we need ATP to make that happen. Okay, we still, maybe we don't call it active transport because maybe you're not always going from low to high. You could be going from high to low. So that's why sometimes they don't call it active transport. I don't really care. Just, again, in mastering biology, there was one of those questions. Okay, so then that's it for exocytosis. Now, endocytosis, there's 3 different types. So it's a little more nuanced for endocytosis. The first is phagocytosis. So phago, that prefix there means eating. And then cyto is talking about cell, so that's why it's called cellular eating. Penocytosis, peno means drinking. And not that you would know about this, but maybe your parents, peno, what does that sound like? That you might drink. Was it? Pinot, yeah, Pinot wine. So how you can remember penocytosis is cellular drinking. So phagocytosis is you're bringing things in bulk. Penocytosis, you're doing cellular drinking, it's smaller amounts. So you're just kind of sipping. You're sipping stuff from the extracellular fluid, just bringing in small amounts of it. Now, receptor-mediated endocytosis, This is specific. Meaning these two, phagocytosis, pinocytosis, they're non-specific. They just kind of, like you just sort of bring in, they're non-specific. They just, whatever comes in, comes in. They have a goal in what they want to bring in, but it's kind of like if you're opening a gate in a neighborhood, sometimes other things, like another car could just like come in. It's not super selective. Whereas receptor-mediated endocytosis, I'll show that to you in detail. That one is very selective for what you want to bring in. All right, well, let me show you these in a little more detail. So phagocytosis, here's the food. This also could be maybe like a bacteria, like in a immune cell. Maybe you want to bring in a bacteria so you can like dispose of it. What you'll do, the cell membrane will form these little projections that we call pseudopodia. Anybody know what pseudo means? Pseudo is cap. It's fake. Pseudopodia. It could be. We could say cap podia. Right? It's like it means like sort of or fake. Like so. Maybe you hear like pseudo intellectual. It's like they're kind of like trying to be fake smart. They're using like big words, you know. And then podia. What does podia refer to? Or what's a podiatrist? Anybody know? Yeah, Charlotte. No. The foot doctor. Yeah. So podia means foot. And you're like, where would they even get that? Because if you look at this here, if you like look at it and squint your eyes, don't think about it too much, it kind of looks like a foot, I guess. So it's like a false, pseudo could also mean false, a false or like a fake foot bringing in the material. All right, and then remember ATP, what's not shown in this diagram, to do this, form this structure, you got to use energy to make that happen. You can't just like accidentally do that. ATP is driving a lot of proteins to make this process happen. Okay. There's some paper towels, or if you need tissues, there's some tissues up there. Okay. Oh yeah, and then this food vacuole, this will then meet up. They don't show it in the picture. This would then meet up with a lysosome, and in that lysosome, what's in the lysosome? There's enzymes. What kind of enzymes? Digestive. But like, what would be like? There's a specific name for it, like a fancier word. When you take a polymer, a food's made of polymers, and you break it into monomers, what chemical reaction are you doing?
Like hydrolyzing enzyme?
Hydrolyzing or hydrolytic enzyme. Yeah. Hydrolytic enzyme. So enzymes that do hydrolysis, they add water to the food vacuole to then split it apart into the monomers. All right, I think that's it for that one. Oh, and remember, this is bulk. So we're bringing in a lot of, like, that's pretty hefty, right? That's, you know, that's like a quarter pounder right there. You know, we're bringing in the cell. So, all right, now, if you're sipping on some Pinot, you know, you're just casual Friday night, pop some popcorn, and you're 21, of course, and you drink some Pinot wine, right? Pinocytosis is cellular drinking, you'll hear, or it gulps. It brings in just small amounts. So just smaller amounts of stuff. Remember, phagocytosis and pinocytosis are non-specific. So that's why you'll see like the purple triangle and the green circle, because you're just bringing in stuff. It doesn't have to be one specific thing. Something else you'll see here is the coated pit. So these coat proteins, they're going to use ATP, and basically these proteins will like use energy to like pinch off that vesicle, and so you'll be able to see those coated, those little proteins in that vesicle. Kind of a little random detail, but it is a thing. But yeah, non-specific. Yep, that's really about it for that. All right, last one is receptor-mediated endocytosis. It's kind of a mouthful, but what it does is really pretty obvious from the name. There are receptors. What is a mediator? Like, say you're having some beef with a friend, and you're like, we need to get a mediator. what's the mediator going to do for you and your friend? It's going to mediate. What does it mean to mediate? It's not a medium. It's not going to like communicate with spirits. A mediator is going to like facilitate. It's going to help like direct the conversation. So receptor mediated endocytosis. This receptor is a mediator to help bring these solutes into the cell. So these purple triangles are specific to that receptor. This receptor only binds those purple triangles. So that's why we say receptor mediated is the specific kind of endocytosis. So when you're being choosy, you're going to be specific for those purple triangles. And then notice you also have the same coated vesicles. So those proteins are using ATP to like pinch off that cell membrane. And as these receptors get bonded with the bound with the solute, they'll then kind of start to like they'll then migrate towards the inner part of the vesicle. So between the coated proteins like pinching and then the receptors like pushing inwards, that'll then form that vesicle that will then pinch in and go inside the cell. Now we are going to recycle the empty receptors. Proteins are pretty expensive. You don't want to just like throw proteins in the trash. You want to recycle and reuse them. So once these receptors are done being used, we're going to put them back in the cell membrane to do it all over again. Okay. Should be about it. Now there is one example of this that I want you to know. And that example is hypercholesterolemia. Easy for you to say, right? Hyper, what does hyper mean? Like hyper, what was it?
Hypertonic.
Like hypertonic. Okay, what did a hypertonic refer to? A lot of solute. So hyper, a lot. Cholesterol, what's that referring to? Cholesterol, okay. Emia, what does the base emia mean? So like in medical terminology, there's like leukemia, anemia. What is the common thing with those? What was it?
You have too little of it?
Too, not necessarily too little. It's referring to a specific thing. Blood. Emia is referring to the blood. So put it all together, and this really big scary word's actually pretty simple. It's a lot of cholesterol in the blood. And why that's bad is because atherosclerosis. I did it. So if you have too much cholesterol in your blood, cholesterol will want to like form these clots in your blood vessels, and that's bad because like heart attacks and strokes and stuff like that. So fortunately, in our blood, we have these proteins called lipoproteins that will bind the cholesterol and prevent it from being able to form those plaques. So you have HDL and LDL. You don't have to know the difference between the two. LDL will be called the bad kind, and HDL is like the good kind. The D stands for density, where the density is referring to the density of protein. So if you are low density, that means that these lipoproteins have a lot of fat and not as much protein. High density, a lot of protein, not as much fat. Don't need to know that. Getting to the point of what I want you to know is there are receptor proteins. So let me jump to the picture now. There are these receptor proteins here, like the goofy looking purple things on the cell membrane. Those receptors will bind to LDLs. So these LDLs, inside these LDLs, it is bonding the cholesterol. We want to get the cholesterol out of our bloodstream so it doesn't form plaques, and we need that cholesterol to put it in our cell membrane and to make, certain signaling molecules, right? So we want to bring those in the cell, but we have to do receptor-mediated endocytosis, right? Here's our receptor. It binds to the receptor, and then we can bring that cholesterol into the cell. This is how it normally works. But there's certain genetic illnesses where either maybe you don't make enough receptors, so notice how there's fewer receptors in the mild disease, or maybe these receptors, they don't bind the LDL as well. It's kind of like a similar idea of what we see with people who, like with the COVID, like the virus mutating, and the antibodies you might get from the vaccine don't bind the COVID virus as well as it mutates. It's a similar idea. It could also happen in the mild disease. Or in the severe disease, you don't have the genes to make those receptors. So those genes just aren't functioning properly. Okay? So then you would have to take some sort of medicine to help get rid of the cholesterol in your bloodstream, so that you don't get like all those plaques. So anyways, that's kind of an example of receptor-mediated endocytosis, a medical example that I want you to know. Any questions? All right, that's it for 5.5. These are just some review slides. But all right, I'll end the recording.